So, good afternoon. Today we have here a visiting scholar that came for a short visit to the New England area, Dr. Gil Rilov. Dr. Gil Rilov is a senior scientist at the National Institute of Oceanography and Institute of Oceanographic Limnological Research, IOLR, which for those that are interested, we actually have an industrial center agreement. So we have a collaboration between the Israeli center and CCOM. Beyond these topics you're going to see here, if there's anything you're interested at the end, you can ask him about the center, which I asked him to elaborate a little bit. A little bit about Dr. Rilov's background. He did his postdoc at Duke University, and after that he went to the University of Canterbury, New Zealand for a second postdoc. Then he came back, went to the small university, Oregon State University, working with a person that some of you might know, Jane Luchenko, and those should know her. For how many years?
3.5 years.
3.5 years, under the PISCO, P-I-S-C-O, Science Coordinator position. And from 2009 till present, he's a researcher at the IOLR. In addition to that, he has a second position at the Haifa University under Marine Biology Department. His main research focus is on ecology conservation of coastal ecological communities, mainly rocky reefs. Specifically, he studies the biodiversity pattern and processes and how they are affected by the bio-invasions, climate change, and coastal development. In addition to that, also marine protected area, as well as benthic pelagic coupling. Currently, he's working on the shores of the Mediterranean and the Red Sea, and also has a project that is experimentally comparing the effects of climate change between the Levant and Baltic shores. Recently, he started a project aimed to map seagrass beds in the northern Red Sea, and he previously conducted studies on the coast of Florida, Oregon, Carolina, Chile, Brazil, the whole world, New Zealand, and South Africa. And in addition to that, last but not least, he's involved in 2 additional European projects. So further on, here's Gil.
Hi. Good afternoon, everybody. Thanks for staying on a Friday afternoon. This is actually one slide that you won't see because of lack of time, but if you want to ask me later about climate change issues, I'll be happy to share those with you. So this is exactly where my lab is, where my office and lab is. This is the IOLR, this is the National Institute of Oceanography in Haifa. Oops, what happened? Situated in the north of Israel. It's part of the National Institute of Oceanography is 1/3 of 3 institutions that belong to IOLR. One is this one, which is the center, and then we have one in Eilat, this is the Red Sea. It's mostly a mariculture facility, and there's one on the Sea of Galilee, Lake Kinneret, which is freshwater oriented, and it's all under the Ministry of Water and Energy, switched to used to be infrastructure. So it's a government research institute. We do have biologists, oceanographers, biogeochemists, geologists, chemists. So we have the whole variety of people to be with the potential to do really cool marine science. Unfortunately, there wasn't too much collaboration among the different sections, which we are hoping to develop now. I'm also, as Shachat said, a faculty at Haifa University, which is this thing here. So this is Haifa. It's situated on a mountain very close to the shore. This is the map where I did my work, so that's the long list that Shachat mentioned. Mostly in temperate areas, all in the yellow stars and the 2 tropical zones, the Red Sea and in Key West here for my postdoc, the first postdoc. Now I'm here, and hopefully we'll get a chance to do some work around here too. when I started my position at IOLR, I established the Marine Community Ecology Lab because nobody actually looked at the entire system from a process and pattern point of view. So we are really lacking data, or long-term data, on the patterns and processes of the ecological systems in the Mediterranean. All the ecologists always went to the Red Sea because it's much easier to work and it's cooler, coral reefs. But so the Mediterranean was completely neglected. Now we have a group of 9 students in my lab and 4.5 technicians. So it's a pretty big group. Not easy to handle. It's grown exponentially in the last 4 years. But really cool people do a really good job, and we are collecting tons of data. As you will see, just a snapshot of that. Hopefully it will not be too much for you. So we work both intertidally and subtidally. Title of the talk today is Trouble on the Edge of the Sea, an ecosystem phase shift on the Levant shore. And you see that the Levant is really at the edge of the sea. This is just a little piece of the rocky shore here. This is a little tide pool in the intertidal. It's all within the framework of the global biodiversity crisis, which is caused by many things. Specifically, I'll be talking today about the Eastern Mediterranean coast. This is the Mediterranean here. It's, this is one view of the world when the Mediterranean is in the center, but it's really a relatively small sea, but with relatively high diversity. And the Levant is this part here. The Mediterranean is divided into 2 basins, the Western Mediterranean and the Eastern Mediterranean, and part of the Eastern Mediterranean is the Levant. We are situated right here, the very edge of the sea. And we have problems here, and they're growing very, very fast, and I'm going to demonstrate some of those today. We should understand that this is really an extreme environment. So these are several maps of the environmental conditions in the Levant. You can see here, this is temperature, that this is the hottest region in the Mediterranean. It also is the saltiest region in the Mediterranean, and the most nutrient-poor environment. It can be considered an ultra oligotrophic system. It is also very fastly changing. This is a really new graph that was produced this year by a scientist in our institute. Here we can see temperature change over the last 30 years, and it has increased by more than 3 degrees, which is huge. In an environment that is already naturally has extreme conditions. This is evidence from satellite data. If you're familiar with this website, Coastal Warming, that looks at all the pixels around the world in the coastal environment to look at the change in temperature. So this is the decadal change, different times of the year. Over there, it looks like it's a little bit lower than that, but I believe this better because we have some instruments that have. These are actual measurements. Salinity is also steadily growing. This is the Mediterranean wide, and sea level is increasing. It has increased by more than 10 centimeters in the last 20 some years. In an area where the tide is this big, this is almost a third of the tide. So this is again huge. Obviously, I'm trying to understand the biodiversity and community structure and ecosystem processes, and eventually their functions. And final goal from the human perspective is look at ecosystem services too. And all of these are influenced by a variety of stresses, of which I'm looking at part of them. This is, I think, true for almost every region around the world. So recently, we have all these new pressures. We have the biggest desalination plants on the planet. Oil and gas drilling has become huge in the last 3 years. They found all these reservoirs offshore, and they started drilling like crazy along the coast. We were always importing our energy. Now people are discussing about exporting energy from Israel, which is unheard of. All these obviously affect the stability and the resilience of the environment, which makes it very challenging to manage this the system, its biodiversity, and we need to think hard about how to conserve it through, for example, a marine reserve, which means that we have to somehow control all of these other things. A big challenge for the environment. So going back to the Mediterranean as a whole, it is highly susceptible for big changes in biodiversity. Originally, the native organisms there were of Atlantic origin. It used to be millions of years ago part of the Tethys Sea, which was tropical, but then it closed from here and opened later to the Mediterranean, to the Atlantic, and was flooded by Atlantic water and species. So mostly what we have here naturally are tropical, more cold water species that are living at the edge there. Remember? Now, not only that, they're already living probably at the edge of their physiological comfort zone. Now, with climate change and bioinvasions, mostly through the Suez Canal, flooding the Mediterranean by Indo-Pacific species, it's a double whammy here, and it becomes less and less Atlantic and more tropical, both in terms of conditions and in species. Of course, on top of that, you have all the other pressures. So high susceptibility to changes. Now, when we talk about diversity, for those who are not biologists, people think about diversity as the number of species, but this is not really representing the environment very well, because theoretically you can have the same number of species while the community is shifting completely from community A to community B to community C through invasions of additions of new species, but as parallel you can have extinction, so you can have theoretically the same number of species, but very, very different species. This can lead to what we call a phase shift, when the whole system is transformed from one state to another state. The system in the Mediterranean, which I work mostly on, are hard substrate, rocky reefs. Here you have a bathymetric map of the north of Israel. This is Haifa here, and our institute is right there. Intertidally, we have a very unique system, which is called the vermetid reefs. I'll elaborate on them a little bit in the next slide, actually. And then subtidally, we have those subtidal rocky reefs, which are very biogenic, very rich, very complex environment. You can see them here. These are actually petrified dunes that were created when the sea level was lower during the ice ages. We have the similar ones which are creating those. This is basically it, but underwater. So it goes along the shore. When the sea level was 100 meters lower, they were created on land and they were flooded. And they create very big reefs. So these are those vermetid reefs. Vermetids are actually sedentary gastropods that are aggregating and are cemented together to create these elevated rims at the edge of the platforms. These tiny holes here are actually the openings of those snails. It's hard to think of this as snails, but this is what it is. Many organisms in the seas, are sedentary. They don't have to move because the food comes to them with the water. And they create these. They basically stop the erosion right at mid-sea level by creating a crust, because this is soft sedimentary rock, which creates a very big intertidal in an area where the tide fluctuations are very small. So these can be tens of meters wide. So it's a very unique system. Unfortunately, and these snails can be considered by ecologists. They call them ecosystem engineer, because they engineer the environment. They change the abiotic conditions for other species. They create a unique system. This is the name of the species that is creating those reefs. Unfortunately, we have very limited data on community structure, at least until recently. And we have very limited data on ecological processes, such as species interaction, benthic pelagic coupling, and so on. It is also considered to be highly exposed to human impacts and a vulnerable habitat by the EU protocols. You can see the distribution of those reefs. This is in Sicily. They only occur in warm temperate water where you have sedimentary rocks that can be eroded. These are corals that we took from those reefs. You can see here, this is at the edge, the sea edge of the seaward edge of the reef. And this is distance from the edge. You can see this is all biogenic. This is all made out of those vermetid gastropod. And the biogenic portion of the reef is becoming smaller as you get away from the edge of the sea. This can be probably 1,500 years old, this core, okay? You can do a lot of things from this, but I'm not going to get into that. Subtidally, the reefs are also very complex. They are highly biogenic. And so on top of the sedimentary rock, you have a lot of buildup, of biogenic buildup made out of shells and corals and things. And this is just an impression how they look like underwater. Now, these subtidal reefs also have not been studied in the region at all. This is a recent publication in PLOS One, led by Enrique Sala. You can see all the study sites of the reefs that they've studied. And you can see a big hole here of all the eastern and southern parts of the Mediterranean. I'm slowly starting to fill this hole. We've done a lot of surveys in Israel, and now in this last June, I took a team and we went to Cyprus and to Crete to do some intertidal and subtidal work, to do some comparative work. So eventually I'm hoping to at least fill this area here. In this review, in this work, Salah described different types of habitats that I think can be mapped using technologies that have been developed here. Areas that are fully protected with a lot of fish and a lot of canopy algae, mostly brown algae, and then areas with not a lot of fish but a lot of algae, and then turf areas with only very shallow macroalgae growth with usually it's red algae, very turfy-like, you know what turf is, and there's some areas which are completely barren. So they have distinguished within these four and talked about conservation. I'm not going to get into that. This is just an impression of how it looks like underwater. Again, in Israel, you can see the water can be very clear. You can have a 30-meter visibility some days. Again, I think very useful if you want to do even mapping from above the water. I'm not going to talk a lot about mapping today, but you can. Well, I think this is a very visual talk, and you can get an impression of how things look like and what can potentially be done with that. Here is another photo. It's almost like some tropical area. Some of the diversity. These are macroalgal canopy, which are very very seasonal, and they're very patchy too. So my ambition when I started this was to talk about change, and mostly focusing on invasions and extinctions. But before we want to talk about change, we need to see what's there, and we didn't have any data on that, any consistent data, any systematic data. So when I started my job, I set up with almost no money a monitoring program, still with not much money, but a lot of motivation. Basically involving working on your knees with quadrats. And so these are the intertidal surveys that we did. We have 4 zones at the edge, the center, and 2 at the back of the reef. We have 11 sites along the coast, what we do annually, and 4 sites, the red ones, that we do seasonally for biodiversity. We've done more than 200 of those transits already in the last 4 years. We also put temperature loggers, tidbits underwater on the platform, so they're partly exposed, partly immersed, depending on the tide and the waves, and one on the rocks to measure air temperature above the water. These can produce really nice time series, which can teach us a lot about immersion and immersion just by comparing the temperature of the air, of the water and the platform and the air. So with the temperature of the platform, the blue line is matching the one end of the water, you know that it's underwater, and when it's more following the air temperature, then you know it's out of the water, and you can start learning about the dynamics of the system. This is a photo from a kite that we took of one of our study sites. My lab manager is also a kite surfing instructor, so he brought up his kite with a GoPro, and we took a photo over one of our study sites. I think I suggest it as a tool for mapping. This is where one of our temperature loggers are situated. It's not too bad, huh? You have to just adjust it a little bit. This is a time series of the last 3 years of the temperatures. Ignore the black ones because I don't trust the air temperatures so much, but you can see here that the 2010 and 2012 were the hottest on record, actually. But it fluctuates. 2011 was a little bit colder. This year summer was a little bit colder, but look at the numbers. Almost 32 degrees, the water temperature. It used to go up to 29, so we know that things have changed dramatically. We also take a lot of other measurements, so monthly We do all this other stuff. We take physical data, these 4 core sites, and we do nutrients. And this is just an example of a time series of nutrients in the 4 sites. I don't want you to get into this so much, but and we also measure other things like recruitment of larvae of mussels and barnacles, and eventually we hope to tie this to oceanographic data as currents and things like that. Underwater, I don't call it a monitoring program yet. It's mostly surveys, but we've done quite a lot of those. They were mostly initially focused here around this. This is a marine reserve in the north of Israel called Achziv, which Shachak is very interested in this beginning collaboration on this area. So this is a reserve, and these tiny islands inside the reserves, and these are 2 tiny islands outside. And I compared the 2 of those areas. I also do work. This is a paper park. It's a reserve that is not being preserved, unfortunately, but I'm collecting data. And for another project that involved an ecosystem environmental impact assessment of the expansion of the Haifa Bay port here, I did a lot of diving on these reefs here. So this is more of the areas that I've been covering, and we worked along 30-meter transects, doing a lot of stuff. We are counting fish and sizing them, and on the transects, we are counting invertebrates. We are also taking photoquadrats to estimate the percent cover of algae and macroinvertebrates and also substrate. We also did, for 1 year, we collected the live rock and brought them to the lab and took measurements of percent cover, and then eventually broke them into pieces to take out all the infauna, all the stuff that lives inside the reefs. And then we developed really a bank of biodiversity photos, and we also take samples for genetics. I'm not going to get into that. So we have, I think, quadrupled our information about the biodiversity, and we did everything quantitatively for this region. We also started doing biomass, because this changes a lot over the year, and between sites, some sites can look like this with a lot of canopy, and others can be pretty barren throughout the year. And the idea is really to look at the variability in space and time, both seasonal variability that is repetitive, and then but sometimes it can be a deterministic change, like for example with climate change. Spatially, it can be either a gradient that we're looking at, or it can be very patchy based on the local conditions, and you can potentially have an interaction between time and space. Some areas can change very little, and other areas can change dramatically, depending on many different things. Just to show you some data, so biological. These are the sites from north to south. This is intertidal data of just the number of species. I told you that's not necessarily the most indicative parameter, but it's also telling us a lot from the edge of the platform, right there, and the center of the platform. So these are just the number of species, but you can see, for example, this is just to show you what kind of information you can get. You have a higher diversity at the edge, the red bars here, than the center, but the center is much more fluctuating, higher variability among sites. But the edge and the center, I'm not going to try to explain that now, but I have some speculation on that. Again, all of these I think can be also measured spectrally, and this is in the tidal, so even during low tide, if you fly over and you have a good camera and you know how to calibrate color to species, or at least some taxonomic level of identification, then you can do a lot. So another type more complicated analysis. I know most of you are not biologists, but this is a crash course, 1-minute course in multivariate analysis of biological data. It's actually very simple. The idea is that you have, for example, different sites, and you have different species, and you have a value, a number of, or cover, or number of individuals per site per species. And then you can create a similarity matrix by looking at all of these combinations together. And basically, this is the percent similarity within the different sites, and then you can throw that on an ordination. And each dot here represents a site, and the distance within the sites represents a similarity. So you can already see that these 3 sites are very similar to each other in the structure of their community, which are very different from these sites. And this site, for some reason, maybe it's polluted. This is all imaginary data. but this site can potentially be polluted, and that's why it's very different from all the others. Okay. I'm showing this because I'm going to show you a few slides with this kind of data, but it's very intuitive. It doesn't have any dimensions. It's just the relative distance between the sites. So here's one of those. It looks very complex, but it's actually quite easy. These are the 4 core sites here, and the different colors are represented here, the same colors. And these are for several seasons, already 2 years of data. And you can see that most of the sites are clustered together. They are more different from each other than they are different among seasons, although there is a seasonal signal, which I'm not going to put much attention on now. But you can see that they are different for some reason. We don't know what is the reason, but they are clustered very nicely together. Okay? And this kind of data you can produce for many different things. You can do it for time, for space, for depth, and so on. We can also, now we have several years of data, we can look at time series of how species are doing, and you can see some seasonality for this species. This is a red calcifying species, Jania rubens. It's mostly a summer to fall species, so that's where you have the highest abundance. This one is also, it's a brown algae, it also has seasonality. This one is a different seasonality. It's more of a winter-spring species. So you can have different species, different colors at different times of the year. So this is kind of stuff you can get from looking at the same site over and over. Again, this is again one of those multivariate analysis graphs. These are names of the sites, okay? So you can see how these sites are similar to each other more than these guys are quite different from them. These are more southern sites. And the bubbles here represent the abundance of the species. And you can see how, for example, this species can be very patchy. In some sites, it's very abundant. In other sites, you can hardly find it. And this species is also patchy, but in a different way. And you can go by species and see where it's found, and then try to explain why it's there or it's not there. Okay?
Is the abundance percent cover?
This is percent cover. And this is the same for invertebrates. This is an invasive mussel from the Red Sea. And you can see that it's not invasive everywhere. Some sites don't have it at all. At the edge, this is actually it's the platform center. You can see here the mussel bed. All this black stuff are mussels, okay? But it's not everywhere. And this is one of those vermetid gastropods that build the reef. And you can see also that it's very patchy. It's abundant in some places, but completely absent in others. In this case, mostly the southern sites. All this, again, I think information can potentially be translated or measured remotely, maybe not at the species level, not these guys, you couldn't remotely sense them, right? But other things can potentially be sensed.
All the areas are the same size, or each sample is 1 by 1 meter, or is it different?
These are 50-meter long transects with 15 quadrats along them that are randomly placed.
Oh, okay.
You don't sample the entire vermeter, but you randomly place the sampling units along the transect. Similarly, you can do that with subtitly. These are 10 habitats, sometimes different depths, sometimes different areas underwater on those reefs. Again, similarity analysis. You can also do a lot of statistics on that, which I'm not going to get into. And then you can see, for example, these are the 4 Haifa Bay sites, which are relatively similar to each other, but different from the ones that are here on the Haifa Head region. Again, this is all based on species data. Underwater, you can see that even within a site, you can have a lot of patchiness. This is just to exemplify how dramatic that can be. This is a macroalgal bed, and see, it looks like somebody came with a lawnmower and cut the grass. This is probably something biological, which I'll go back to later. But this can, I'm sure, this can definitely be done underwater by surveys, and we've done that. And here you see a patch around this reef, where you don't see the macroalgae. I think this can be done remotely too. This is what I call probably the grazing land caused by this invasive herbivorous fish. I'll go back to that later. And I think this can be mapped remotely. This, for example, is another. This is an invasive algae. It's a red algae, so it probably has a different signature spectrally, and which is very patchy. But here in this patch, it is almost 100% cover. You see, these are the quadrats that we are doing. So what I showed you so far is how we measure things. But part of the reason is to look for patterns in time as well. I'm mostly talking about species additions and species deletions. This can be collapses, extirpation, so a local extinction or global extinction. Right? So just to exemplify the sort of data that we have, so these are those invasive mussels that are very patchy, and this is just to graphically show you or visually show you how patchy they are. So all the red dots are where you have a continuous mussel bed, and where you have yellow or orange dots is where you don't have mussel beds, or they are very, very small. And you can see that there's no north to south gradient, but it's very, very patchy. One of the things that I'm trying to understand is why is it patchy? Is it something very local? Is it something oceanographic? To do that, I do also some experiments. I'm just going to show you. This is a setup. I'm not going to even show you the data because we don't have time for that. But for example, one of the things that can lead to a patchiness is differential recruitment of the larvae of the mussels to different sites. So for that, we use artificial settlement collectors that are now present all over the world. I use them in New Zealand, in Oregon, in other places. I didn't invent them. These are basically pot scrubs, what the mussels are love to settle. But you can then put them for a day, a week, a month, and then bring them back to the lab and see how many mussels settled in. And I also looked at predation, because mortality can also explain the patchiness, okay? Differential mortality.
Do you standardize the space?
These are exactly the same everywhere. They are manufactured.
And so you just compare what's in each sampling unit?
Yeah, I have the data, but I'm not going to show it because we don't have time. I'm just showing you what we are doing and to try to explain the processes that lead them to the patchiness, for example. So for the predation, we transplant the mussels on plates to serve them for predators, but then we put them as control, as open, and then we can protect them from different types of predators using only a roof or a fence or a roof and a fence. So this, for example, is protection from fish, and this is for protection from creepy crawlers that they cannot feed from above. Again, I'm not going to show you the data because we don't have enough time. We also try to see what this mussel is doing to the system, looking at how it changes the diversity. There's a student that have done that. So we're looking at areas with mussels, what other species are there, and without the mussels. Again, no data. Subtitly, we have a lot more invaders. This is a 50 by 50 centimeter quadrat that was taken here on the Haifa head. If we had time, I would test you, but we don't, so I'm going to point you to the invaders that are found here. One is this is an IndoPacific sea cucumber, about this long. Here we have this oyster, it's called Spondylus spinosus. These are really huge. Oysters, very thick, very, and sometimes they can create reefs. I'll show you that in a second. We have a very abundant snail here, and two invasive macroalgae, Galaxaro, which I showed you earlier, the patches, and another one, Codium. We have two species, so I don't know which one is this exactly. They're all invasive. There are probably several others that are hiding there. So this is only 50 by 50 centimeter quadrat. Okay? But to really blow your mind, this is the relative abundance of bivalves, gastropods, and fish on the reefs. And everything in warm colors, browns, red, pinks, is invasive. You can really get an understanding of what's going on here. Every 99.9% of the bivalves that you count at the water is invasive. Mussels and oysters and whatever. Okay, 95.5% of the gastropods that you count at the water, I'm not talking about species, I'm talking about individuals, are invasive, and fish can vary between 30 to 70%. So we are really witnessing a huge invasion, and they are ecologically important because they are where are the native ones? Nobody ever counted them in the past, so we don't have anything to compare, almost nothing. I will show you some data. And this, for example, these are those oysters, and look at this reef that it is creating. All of this is made out of live ones. These are some of the dead ones. They are dead probably because of this guy here, this octopus that enjoys eating them. But you can see how it adds to the complexity of the reef. So this is not this is another ecosystem engineer. It completely changed the three-dimensionality of those reefs. You can see here, this entire reef is made out of those things. In terms of fish, we have a lot of species. Some of them, we never had true herbivores in the Eastern Mediterranean fish. So, and these guys are doing a lot of damage. I'll show you that in a second. These are nocturnal planktivores, which we never had in the Mediterranean. We didn't have this kind of species. So did new functions too. These are the algae. They can be sometimes be very abundant. Just an example what these two herbivorous fish can do, they can transform fleshy forest into a turfland, turf So all these fleshy, tasty macroalgae, brown algae are being eaten, and we are left with a much poorer environment with only those red calcareous algae, which those fish don't really like to eat. This is, I think, what is happening here. This is a flat area, no texture. So no hiding place for the fish, but the fish, there is a crack here where the fish can live, and they come out of this and they feel safe to go this far and eat. And if that's probably where they turn back because it's too far for them from this, and they eat away until they, get too scared and come back. So this I think is a grazing land.
So Gil, did you have true calcified reefs there? Because in a lot of places around the world, right, there's this switch between calcified reefs to algae, and that's because the herbivorous fish are going away. Whereas you seem to have the opposite, where you have this macroalgal dominated system that's becoming kind of a turf.
Yeah.
Did you know what you had originally?
No, we don't have any. I think it was mostly we did have those brown algae as a dominant feature, and we're losing that now. Most of the area looks like this on our coast, and it's true. There was a paper by Enrique Sarra that did these experiments with the cages, which we have repeated later. So when you put a cage on this kind of habitat, immediately those brown algae start growing when they're protected. So I imagine that it was mostly like this. Now we see it very, very patchy. Again, something that you can map easily, I think, underwater. And maybe from the air too. Some examples, so I've talked a lot about invasions and just demonstrate how pronounced this phenomenon is. Let's talk about collapses. This is something that nobody have really looked at in the Mediterranean. I'm talking about mostly, so collapses can be occurring for many different reasons. You can be overfishing species, you can be destroying their habitats, you can have invasive species eating or competing with native ones to extinction or at least collapses. You can have pollution that is causing collapses of insensitive species, and obviously climate change that change the environment totally on a regional scale to make the conditions unhospitable for the local species, right? So very little about fishing-induced collapses. And the way you can do that is to look at what happens in a fished and non-fished area. We hardly have any such areas except for this one little reserve in the north. Normally what you would do is to, if you want to see the effects of fishing, is to use a bucket design. This is, for example, you can compare two sites before you put a reserve, and obviously both of them are pretty depleted in numbers, and then you protect one and leave the other unprotected. Supposedly you should see more fish and bigger fish inside, and then you can see what it means to the habitat. So in 2010 we went for a little excursion in the Oceanographic reserve. These are those tiny islands that around the area where Shachaf is also interested in. So this is the protected area. These are those tiny islands. You wouldn't call them islands really. It's just a little reef sticking out of the water. We don't have any islands as for us. These are islands, and this is outside of the reserve. And what we did only after, we don't have any before data. So we went on a mission and we sampled quite a few sites for both fish and invertebrates, and we did photo quadrats. This is a three-dimensional depiction of those sites. And it was a big team, 5-day diving. And we also even took measures of currents, and we used CTD to measure the quality of the water. You can see how transparent the water is, right? Very clear. This was in May. This is just to exemplify what kind of data you get and the effect of the reef. So here we're talking about 4 native predatory fish that are commercial. And these are the 2 invasive herbivorous fish. What stands out really, and these are 3 different depth strata. This is the most dramatic difference. You can see the very shallow waters, you have many more of those commercial native herbivores than outside of the reserve. The rest of the depths, not so much, only because it's further away and it's more difficult to protect them. There's also more of those invasive herbivores inside the reserve compared to outside, but not as dramatic. This is not very surprising. What was surprising was that the benthic cover was very different. Outside the reserve it was mostly turf, and inside it was mostly the fleshy algae, at least in the shallow water, which this came as a huge surprise to me, which suggests to me that the effect of the protection trickles down the food web. And somehow I can go into speculation, but I don't have time for that. You have much more of the native, original communities there inside the reserve. So it's not just the fish, everything else is being affected by the protection.
Maximum height to become a canopy?
No, it's not very big. It's not kelp. We don't have kelp in Israel, but this can grow at the peak of the season. You know, sargassum and other things can to be this big, but it's normally shallower than that. These are those photo quadrats before they were fixed and straightened. You can see this is inside the reserve and outside. It's a huge difference. Again, I think easily mapped. And this is expressed from those photo quadrats. You can see that the communities inside the reserve are quite different from inside of the reserve. This is another guy. There were quite a few more octopuses, for example, inside the reserve than outside. So this was an example of how fishing is affecting. So, and most of Israel now, it looks like outside of the reserve, because it's outside of the reserve. It's not protected, and we have very few of those commercial fish. But it shows us also the potential of this reserve of how a healthier system, although we have the invaders, should look like. But now I'm going to switch to non-harvested related population collapses that are not easily explained. It's not somebody's picking those. As I said, we have very few past data, almost no historical data, so no transits, almost nothing. But here are 3 examples of the little data that we have that demonstrate that we are losing species. These are those dendropoma, those vermetid gastropods that build the rims at the edge of the reef entirely, this ecosystem engineer. And during my PhD, I was lucky enough to measure. That was not the focus of my PhD, but I did put a transect to measure how many live ones were there, percent cover of live vermetids, and it was almost 50% in this site here. But I already noticed that in some areas you don't see that very well. You don't see the very pronounced rim. In the last 5 years that I've been monitoring now continuously, we've hardly found one individual that is alive. This last year we found a few. So ecologically, this species is extinct. What does it mean to the system? You can imagine. I'm going to talk a little bit about that. This was the main focus of my PhD. This predatory whelk snail. This is about this big, so it's not very small. It used to be very abundant. In the past, in historical times, they used to use this to extract a dye to paint royal cloth. So there must have been millions and millions of them. But they stopped up hundreds of years ago or thousands of years ago, so that cannot be the reason. But during my PhD, I didn't see any recruitment of juveniles to the population for 5 years, and I actually wrote a paper that I expect that this species will go extinct. Back then, it was still quite abundant, and lo and behold, so this was during my PhD. It was starting to reduce in numbers. I couldn't really show a real trend, but for the past 5 years, we don't find any larvae individuals. And this used to be a very important predator. And here's an herbivore. Urchins, I'm sure you're familiar with, were very abundant along the Israeli coast. If you ask divers 20 years, 30 years ago, it was, when you went into the water, you need to be careful not to step on them. There was one master's student that did the work in the Oceanographic and Achziv area, where those MPAs, the protected areas, these were the abundances per square meter back then in the 70s. I did a few measurements in the beginning of my PhD when I didn't know what I'm going to do. I threw a lot of quadrats everywhere. So these are different sites, but these were the numbers in the mid-90s. This is what happens now. It's very, very hard to find urchins now along the Israeli coast. So they have completely gone. And they are very important herbivores, supposedly, and we lost another function. Those herbivores are now gone. So we don't know the trajectory of those, because we didn't have any monitoring. And we don't know if it's because of a common driver or different drivers, and certainly what are not the ecological effects of those, but because these are such important ecologically, the species are such important ecologically, they must have dramatic effects on the system. This is, again, just to exemplify, so this is 96, still all these holes are alive. This is an operculum, this is the cover of the snail here. And in 2010, when I started looking at the rocks, they were all dead. All those rims were dead. So we basically lost this species. Ecologically, we still find a few here and there. We even wanted to see how quickly those rims will now start to, we expect them to start deteriorating. So we developed our rim meter, which measures the height of the rim along the transects. As I said, we just last 2 weeks ago, we found a few live individuals, which was very exciting because we haven't seen any for the last 3 years, but they are so sparse, so I don't think that means anything ecologically. Why are these findings of collapses important? First of all, this is the first data to show on the large scale multiple collapses of abundant non-harvested species from several trophic levels on warm temperate open coasts. No, not estuaries, not where you're harvesting, not polluted areas, open coast. It also is a strong new evidence that the Levant is going through an incredible restructuring of its coastal community, not just through invasions, which everybody talks about, but also by losing species. Finally, I think it also serves as a warning sign for similar population collapses that may soon occur further west in the Mediterranean. What I think we are seeing here is the ecosystem phase shift from a temperate Atlantic ecosystem to a tropical Indo-Pacific one through invasions and extinction, and I think it's also aided by climate change because it makes the system much more hospitable to those invaders and much less one for the native species. Is this a zero-sum game because we're losing species and adding species? Are we mostly witnessing species replacements? Are we seeing changes in traits? This is, if you were mostly biologist, you would understand what I'm talking about. But I think common sense also, you can understand this conceptually. I would say no. We are losing species with no replacement. We have lost an ecosystem engineer in the intertidal. We lost a predator. We don't have any replacements for those. We have gained a filter feeder, those mussels, which we never had in the past. In the subtidal, we have gained an ecosystem engineer, those oysters. I don't know if we ever in the past had something native that was so dramatic. We lost predators by fishing. We lost an herbivore by those urchins, but we gained those fish that are decimating all the algae. And I think overall, the impact is negative. We are losing biomass because the fish are much more active, they are much more mobile, and they are eating the algae much more efficiently. We also gained all these bivalves, so we added, I think, more filtering to the system. So now, in the last 2 years, I want to understand why we are losing those species, and I'm suspecting it's climate change because what I said, I think this system is very sensitive, and I think the species, they are very close to naturally to their comfort zone. So I'm looking at the effect of sea level rise and consequences to biodiversity, temperature rise, and effects on population collapses and species species and communities, effect of ocean acidification, effects of extreme events. This is something that people are starting to realize that it can be one extreme event that can have effect for a very long time, and eventually we want to try to develop projections for the future. I'm not going to get into data. I'm just going to show you what we're doing, and I'm going to end with that and open it to questions. So one project that we have is to look at the potential biodiversity shifts because of the effect of this ecosystem engineer, the Dendropoma that builds the platform, is now extinct, together with sea level rise. And I've showed you that sea level is rising. What we see, if we put quadrats at the platform level, 0, you see much higher diversity than anywhere else, a little bit lower on the shore, 10, 20, 50 centimeters. I don't know why, but certainly we have much lower diversity, which means that as sea level will rise, we potentially will be witnessing reduced biodiversity. Again, we want to understand the mechanism why we see lower diversity, and we are developing experiments to do that. The vision might be that until recently, this is how the reefs look like. The red stuff here is the Dendropoma, those vermetid gastropods. Now they're gone. Sea level is losing a little. And I think we will be starting to lose species and also structure. We're going to lose some of the complexity of the reef. And maybe in 500 years, it will look like this, with very few species. So one of the things that we're doing, this is within ISF. It's like the National Science Foundation, ISF. First, we started measuring the height of those platforms to try to see how much are we going to lose if the sea level rises by another 5 centimeters, 10 centimeters, 20 centimeters. So we bought this laser total station thing that you can measure. Then you can go with this rod. It's electronic, and you measure the height at different levels. You can see these are the 4 core sites that we have here from north to south, and you can see that their height is different. So they may be affected differently and at different rates. This is my PhD student who's working on that. This is an adventure that we did. This is something we started doing with this guy from the Technion. He has this LiDAR, this land-based LiDAR. We went to one of our study sites and mapped at a millimeter scale the topography of the rock. We haven't gone any further, but my vision is to maybe put this thermal map, and maybe on top of this biodiversity map to start to see at a very small scale how diversity changes. And then you can model sea level rise and see what might happen. Okay, so this is the future. We also want to mimic sea level rise by transforming the diversity from this level to the lower level. We were thinking of 2 different ways to do it, either to take big cores and move them and transplant them lower on the shore, or develop a community on an artificial substrate that will let it develop naturally and then move it lower on the shore. These are all very different, very difficult technically, so we're still working on that, hopefully this year. Another project is to try to understand why the collapses happen. Some of the species we don't even have enough to do any experiments with. Urchins, we actually have to bring Mediterranean urchins from the Red Sea facility where they grow them, not in the sea, not in tanks, to do experiments to understand what are the reasons for their collapses, and also what does it mean ecologically. So we've done an experiment where we exposed, I suspected that it's just too warm for them in the summer. I told you that it's already risen by 2 or 3 degrees in the peak of the summer. So we put them in tanks in ambient temperatures, ambient -2, which resembles what they had 20 years ago, and ambient +2, and look at their survivorship and other things. This is just to see, this is ambient +2, they immediately died. Ambient -2, they were quite okay. This is survivorship. And ambient temperatures, they died quite fast. These are adults and these are juveniles, but they basically had very low survivorship already ambient temperature, which means that they indeed are dying of heat in the summer. They just can't make it through the summer, through the peak of the sun. And I think this is the main reason why they're dying. It can also be competition with those herbivorous fish. I don't have time to get into that, but we've done some experiments on that. With cages where we had fish and urchins and just fish or just urchins and looked at their functions. And this is the most ambitious project that we went into now. This is the work that we're doing in collaboration with Gil Rilov in Kiel in Germany, when we look at climate change effects in the Levant and the Baltic. This is my PhD student. This is Martin Voll, my collaborator from Gil Rilov student. We are looking at species responses, single species responses to temperature and pH stresses, and then we are going to look at interactions and how those are affected by pH and temperature. And the most ambitious one is to look at the whole community, impacts on diversity and ecosystem functions. So the idea behind it and the hypothesis behind it, this is the Baltic and this is the Mediterranean. The Baltic is much more naturally fluctuating in both pH and temperature and also salinity, which we're not looking at, than the Levant. So we are suspecting, the Levant is relatively stable compared to the highly fluctuating Levant, and we're suspecting that because of this difference, the Baltic species will be much more tolerant to fluctuations, climate change-driven fluctuations, than our Eastern Mediterranean. So the idea, in terms of functional response, if you're looking, for example, at temperature, this is just a fictional cartoon of that. The performance, this is a performance curve. We expect that the Baltic species will be have a much wider tolerance range than the Mediterranean one. Okay, does that make sense? Because they are much more sensitive. Okay. So what we've started doing is to look at species by species. We've developed this. This is what we call the microcosm. The Germans, everything is fancy there. They have those thermobaths that they can buy off the shelf. They're very expensive, so we have to develop our own thermobaths. But they work very nice. It took us half a year to just calibrate them. But now we can cool the water and warm the water as we need. So we have now 11 temperatures from 15 to 35 at 2 degrees intervals, and we can produce those curves. I'm not going to show you that because we don't have time. If you're very interested, I can show you at the end some of those curves. And to look at the entire community, we have built a mesocosms. So again, the Germans have massively fabricated mesocosms. Ours are cheaper, but only from the outside. So we had to buy off the shelf. These were actually specially designed for them, but we bought these things and we covered them by styrofoam to make them more insulated. But all the controllers and everything this is state of the art. So we didn't compromise there for quality. So I think they'll do the job. This is still under construction. These are custom made coolers. What is unique about this system, you can see here, this is how it looks like now. Hopefully a little bit better now. This was from a week ago. Still under construction. What is neat about this system that we don't have constant temperatures or pH. We are measuring what's happening at the sea, and we are matching that in the system, and only add the delta of the temperature or reduce the delta of the pH. So we are following the natural fluctuation. We also have, it's an open system, so we have continuous water supply from the sea. So it's an open system. We can get the larvae of the stuff that can come inside, and we can look at all the processes inside. So we can impose extreme events, for example. We can program to cross 3 days of very high temperature and see what that does. So it's a very flexible system, very versatile system, and I hope it's going to work fine. It has a lot of potential. The idea is to have two different treatments of pH, two different treatments of temperature, and then the combination of them. So only ambient, only changing temperature, only changing pH, and changing both. And what we're going to do here, we're going to put live rocks, so the entire community inside, and look at the change in diversity and functions, for example, photosynthesis and other things. And we also, as I said, looking at extreme events. I'm not going to get into that. This is working with climatologists and look at the ecological response of that. The vision really is, and I'm going to end with this, is to look at all these, to explain biodiversity by looking at different levels of observation, from the very small quadrat size species interactions and recruitment to benthic pelagic coupling, and then into ecosystem functions, so looking at a much larger scale, and how the bioinvasions and climate change affect those. And for that, you need to do experimental ecology, you need to do physical oceanography and modeling, which I hope to get into in the future when we have more information. And this is the real future, hopefully. I'm starting with quadrats on the shore. I've started doing some subtidal work, and then eventually I hope to also incorporate into that remote sensing, you know, this kind of stuff. Thank you very much for listening. All this work cannot be done with a really good team of people in my lab, students and technicians in the present and the past, and quite a bit of funding that I was lucky to get in the last 4 years. And if you have any questions. Just to explain this picture, this was one of my technicians just left a few months ago. This is his 40th birthday, celebrating with balloons underwater.
Thank you very much. Before just a question, just another comment. The IOLR, there's other researcher. Hopefully we can get some others over here regarding coral and chemistry, and there's other different ocean mapping aspects. So I encourage people at CCOM that are interested to see what is done in IOLR to go to the IOLR website. And maybe we can convince, if they're coming to New York or DC, to stop by here and give a presentation. So now questions. Yuri.
Gil, what about other parts of Eastern Mediterranean? Probably they have similar investigations of species in Greece, in Adriatic.
Unfortunately, very surprisingly, very little.
Very little. I was just wondering if they have similar tendencies, or are you, you as a country, more affected by the Indo-Pacific invaders through the Suez and
So people in those countries are documenting invasions, so they tick any species that arrives, but they don't look at they don't do any monitoring or anything quantitative, unfortunately. Most of them study soft benthic environments, not so much rocky reefs. In the whole of Greece, there's very few people who do that. I had to go to Crete to sample their sites because nobody does that. And similarly in Cyprus, I was surprised. But people are monitoring at least the arrival of species. We are certainly the ones that are getting them first. For most species, not all. Sometimes, especially if it's probably, if the vector is ballast water through ships, they can land away. So their first landing site can be not necessarily Israel. But the ones that I think that are entering directly through the canal, first land in Israel and then they spread. And they can, from there, they can spread either by swimming or releasing their larvae or by ballast water. So I think there are multiple vectors going on there, and it's very hard to separate them.
I have a question from a geomatics perspective. You said the zero net thing about biodiversity over the same area. If I survey an area, let's say 10 kilometer by 10 kilometer, and I find 2 patches of a kilometer by kilometer, but now shifted, but they're still in that big area of a 10 kilometer, but they just shifted. Does it, from a biologics perspective, did it change, or is it zero net?
If they shifted, if it's the same species that shifted locations, probably not a huge deal. But if it's a different species, it's one patch replaced by a different species, which may have different functions. That might be cool. That might be important on the functional level. Even if it's patches of the same species that have shifted, and that can happen, it can have meanings in terms of functionality. If it's a different depth or a different currents that are going on there, probably less dramatic than switching from those brown algae to coralline algae, an invasive one. So I think that will be, and I have a student actually looking at what are the is there a difference in the diversity in the invasive algae patches versus the native ones? And we are going to manipulate that to see if they replace their functionality, because they're also much more complex than just the bear or rock. But I think they are much less. They are giving much less complexity, and maybe they are less edible. So these are things. These are the processes that we are looking at, where we haven't mentioned, and we are only starting to learn the system.
Derek.
So it seems like your results showed that marine reserves provided some resilience to climate change impacts. Is that compelling discussion of additional marine reserves in the country?
I did not say anything about resilience to climate change, which I think it's true, but this is something I haven't shown. I think what we are seeing is, and I haven't shown that, is that we do have a more intact community because we have more predators and less of those herbivores. Maybe they affect, although there's also more herbivores there, there may be their activity may be reduced because of the presence of the predators. What we also see, and I haven't mentioned that, which is interesting, is that we have less of the invasive species there. And this is something that people talk a lot about, invasibility, and that more intact environments are less prone to invasive species. They don't do as good in more intact environment than this. But I think people are talking about more resilient, that more protected areas will be more resilient to any kind of change. So if it's climate change or invasions or even pollution. Areas that are more disturbed by either fishing or something else are more prone to further disturbance. Okay? It will be very interesting to see if species there are healthier, also in relation to climate change, because they are grazed less or something else.
I was wondering if you think there's any immediate negative effects from those sort of structural engineering additions to the hard surface and also from the filter feeders?
Something that we want to really look at, because this is an ultra-oligotrophic system. The plankton density is naturally very low. So if you add all these filter feeders, they can potentially increase the plankton availability even further if in areas where they are very dense, and they may even clear the water further. People in lakes, invasive species that invaded lakes, have managed to turn a green lake, very murky lake, into a clear water lake. And it even happens in the Gulf, in the San Francisco Bay, where we have invasive bivalves. They are so abundant that they manage to clear the water, which can have cascading effect. It brings in more light. It can increase the growth of macroalgae. So you have a whole cascading effect. We don't know what is happening. Potentially, they are not only adding structure, they may be affecting the water above the reef, but we don't know that. This is something that we want to measure and try to model even. Yes.
Are there thoughts to put any structures underwater to allow the native species or invite invasive species to come into the waters? Like building some cages or building some structures?
I think the evidence in other places shows that artificial structures are more prone for invasions than natural structures. If you open a space, it's normally those invaders that come in. So artificial reefs, which are used for many reasons, for example, for facilitating fisheries, increasing structure, may potentially serve as a stepping stone for invasion. So we don't know. I mean, we know that on ports, usually you find many more invasive species. That's also because this is the inoculation area, which is where you get the invasive species when they come through ballast water. This is the first entry area, so you can have them propagating them from there. So that's maybe another reason why you have more on artificial structures. So I don't think we are lacking structure here. We are lacking protection. I think that really the solution for many we cannot get rid of most of these invaders, but maybe we can reduce their effect by protecting the environment from other stresses, such as, for example, overfishing. And this little reserve demonstrate that. And I'm trying to really give data to help the nature protection, the Parks and Recreation Authority, give them tools to help promote the development of more reserves along the Israeli coast, and so they're very happy when I show them this data, then they can use it for that. Yes.
A question about invasive species. Is there a hope to try to eliminate the invasive species to return it to what would be considered its more natural state? Or do you just have to live with it? They're there, you know, you gotta move forward. And then how long, if a species is there and it's an invasive species, how long until it's considered a natural species? It's been there long enough, it's no longer invasive.
Yes, these are two very different questions. The first one is, the answer to the first one is in 99% of the cases, you can eradicate them. If there were a few successes, for example, with Caulerpa taxifolia, it's an invasive algae that is very poisonous to other organisms, so they cannot eat it, and it can spread very quickly. In the Mediterranean, it's invaded and it's everywhere. In the Western Mediterranean, nothing can be done about it. In Australia, they found patches of it in one port, and they poisoned the entire port, and they got rid of it. They killed everything else, but they also killed that. But because this one is really problematic, they decided to sacrifice the sport for that, and it was successful in other places. In most cases, especially in the marine environment, where you have the medium, the water that transports everything very quickly everywhere, it's very hard to contain or to remove or contain a species because its larvae will propagate everywhere. So basically, in most cases, you have to live with it, or maybe, and again, I think protection, making native species more healthier by protecting them may be one way to at least keep them at bay, not spread. Again, going back to the idea of having more intact system. Your other question is more philosophical, and people are asking that, and rightfully so. When does a species that is invasive is considered native? I don't have a good answer for that. Species that invaded 500 years ago in Brazil during the slave trade that's where I worked in Brazil, where they have this mussel. People really love to eat it, but it's still considered invasive. An old invader. It certainly changed the system. What can you do about it? No, not much.
Actually, there is an answer for that.
What?
There is an answer for that. Someone said that invasive species stop being invasive when they come in a balance with the other species. When they stop increasing or decreasing and come to a balance.
I don't know about that.
Well,
there's different terminology. They call it non-indigenous species, any species that occurs in the system. Invasive is when it's becoming very abundant, and also usually some people say that it's when it's harmful to the system, or when it changes the system dramatically. But any species that occurs in a new system has some effect. It interacts with something, but if it's in very low abundances, maybe it doesn't do much. I don't think anybody made a rule. I don't think anybody can make a rule of when species it cannot be called invasive anymore. Invasive species are normally ones that have been transported unintentionally or intentionally, but by humans. Because there are natural expansions of species, when things are now warming, water are warming, things are expanding, species are expanding their distribution or retracting their distribution. This normally is not considered invasive, although it is. They are invading new territories.
But we are talking about our lifespan. And when we are talking about like longer periods of time, they stop being invasive and become.
Again, I think it's a philosophical question. You're right to have that.
If this poisonous stuff would take over the whole world, that would happen long time ago. But it didn't happen. So that means that there is a natural.
Usually they have a boost and then they flatten and sometimes they collapse too. Yeah.
As well as.
But it still doesn't mean that they're not invasive. Yeah. But I don't have a really good answer for that, and I don't know if anybody. It's really almost a personal opinion or something philosophical. in fact, you know, if we think about the Israeli coast, which was a relatively nutrient poor in species, we have increased the number of species in total. Some people can actually see that as a benefit. We have more species than we used to have. Some of them are very colorful. If they come from the Red Sea, I can show you pictures. They you know, maybe in 300 years, when the weather gets very warm in the winter, maybe we'll have coral reefs. Some people will definitely like that. Yeah. Other questions?
So on that deep philosophical question, thank you very much to Dr. Gil Rilov.